Hello, everyone. Welcome to Origin's webinar on transfection of immune cells. So transfection is an essential technology for delivering exogenous material into a living cell. Chemical transfection is the most commonly used. Later, electroporation evolved to deal with some more difficult cells. So even as the methodology evolved, some of the cell remains to be difficult to be transfected. So immune cells are one of such difficult cells. So although the electroporation has been used, scientists are still waiting for a simple chemical transfection method. So today, Origin team up with Lipocalyx, the transfection experts. We will introduce you to a novel solution to transfect immune cells. So I will leave the story to Dr. Bettina Weber. So in the next 20 minutes, she will tell you more about Viromer transfection to deliver either plasmid, mRNA, siRNA into immune cells. And a few logistic issues. So all of you are muted. If any question, you can use the question box on your screen to type in during the presentation. And these questions will be addressed at the end of the session. We will have a dedicated Q&A session. And all the presentation of this webinar is downloadable. And so whoever attend this one will receive an email regarding the link. So without further ado, here is our transfection expert, Dr. Bettina Weber. Bettina?
So hello, everyone. Thanks for taking the time to listen to our webinar about immune cell transfection. So my name is Bettina Sannemann, and I'm working for the Lipocalyx company, which is a developer and manufacturer of the Viromer transfection reagents. So just a little schedule for today. We will have a look at why the transfection of immune cells is so challenging. We will have a look at the Viromer reagents and how they improve the transfection. We will have a look at the possibilities in immune cell transfection for siRNA and microRNA, and also for plasmid and the new strategy of mRNA transfection. I will explain a little bit how you can change or switch to mRNA transfection, and of course I will summarize everything up. So to really understand why the transfection of immune cells is so challenging, we need to have a look at the process of transfection. So what we have here is a cell, and the normal process that you all know. You take your nucleic acid, which could be an siRNA or a microRNA, a plasmid or an mRNA. So let's say your payload, and you put it together with the transfection reagents, and you form a transfection complex. And these complexes you put on the cells, and then the cell is taken up, the complex, by endocytosis, and then you have your payload in an endosome. So then there are two possibilities of ways it can go. So the first one is the lysosomal way. So there will be an endosomal maturation and a fragmentation of the payload that you wanted to brought inside. The other way, which we are all looking for, is the escape from the endosome. So you have a release of your payload, and you can have your siRNA in the cytosol to go for a knockdown, or you can have an mRNA in the cytosol to go for an overexpression. And you can also have, of course, your plasmid, which will be transported into the nucleus for gene transcription. So the transfection reagent, let's say the chemical transfection reagent, either it's polymer-based or it's lipid-based, it's always just packing your payload. And the cell is taking it up by endocytosis on its own. So it's not forced by the transfection reagent. It's a cellular mechanism, which means the other way around. If you have a poor endocytosis, then you will also have a poor transfection efficiency. Having a look at the cells of the immune system, you have on the one side the lymphocytes, which mediate the adaptive immune responses, and on the other side you will have the phagocytes, which mediate the innate immune responses. So having a deeper look into the lymphocytes, you will see that there are B cells and T cells, you will have the natural killer cells, and you know it better than me that lymphocytes are really, really hard to transfect or even untransfectable. So there is this really big theory that says that lymphocytes have a clear lack in undirected or not receptor-mediated endocytosis, which is, like we learned a minute ago, is the main requirement for transfection. That means for you that all chemical transfection reagents will fail if you want to transfect the lymphocytes, and there's a really clear need to go for electroporation or viral transduction if you work with lymphocytes. On the other hand, we have these phagocytes, which are like monocytes, we have dendritic cells, we have macrophages, and a lot of other leukocytes. And these cells screen the extracellular environment for pathogenic materials, and for that they use the phagocytosis, ingesting the particles that they find. They destroy these foreign particles, pathogens, and the cell debris, which means that phagocytes are potential transfection hosts. So, going back to the picture for the process of transfection and having a deeper look in the normal transfection of the phagocytes. So, what phagocytes do is digesting what they are taking up. So, after transfection, you have your payload in the endosome, and normally it goes in a, let's say, huge portion. It goes the lysosomal way. Things will be digested. For some cells, things will be presented on the cell surface to other immune cells. So this is what normally happens. You will see pure expression rates or pure knockdowns, even though the transfection was working pretty good. So what we provide as a lipocalyx company is a new transfection reagent, which is called the Viromer. And this Viromer is providing an active escape from the endosome. So the possibility that it goes the lysosomal way is much smaller than with other reagents. And so Viromer is very, very fast, providing this escape from the endosome and pushing the whole process of transfection in the direction of the release of the nucleic acid before it can be, or before the fragmentation can be started. And the Viromer, they do so by mimicking the uptake of the influenza virus. That's why they are called Viromer. It's a mixture of virus and polymer, so they are safe and they are totally polymer-based, but they follow the uptake of the influenza virus. And influenza is entering the cell with a pH-sensitive peptide of the hemagglutinin, and we translated that into chemistry. So we have a polymer and with several side chains which are providing the hydrophobicity and also sensing the endosomal pH. And what is happening if you have a transfection complex with your payload and it's taken up by endocytosis, you will have a lower pH in the endosome because of the acidification. And then the Viromer can react to that and becomes lipophilic and could walk through the endosomal membrane directly into the cytosol, and the payload will be released into the cytosol. So with these beautiful pictures on the right side, you can easily see for the HeLa cells as a model cell line. We transfected them with a red labeled as siRNA. So 1 hour after transfection, you see some of these endosomes already in these yellow-orange dots. 3 hours after transfection, there is accumulation near the nucleus, which you can see here. And around 4.5 hours, there's a discharge from the endosome, and you can see these beautiful red clouds, which are mediated by the red labeled as siRNA. So, just to sum it up a little bit, there's a clear and big and loud go for the transfection with Viromer of phagocytes. So you can definitely go for macrophages, you can go for monocytes, dendritic cells, and microglia. And there's also a really loud no for lymphocytes because they don't do this undirected endocytosis. So do not try to transfect B cells, T cells, or natural killer cells. For them, go for electroporation or viral transduction. So this is our product portfolio. We have these three colorful ones who have been our first generation of Viromers. The Viromer blue and green, they are for RNA and microRNA transfection. The Viromer red is a little bit more universal. It's for plasmid transfection, mRNA transfection, and also shRNA transfection. And these silver ones, they are our next generation Viromers, just released a few months ago with the Viromer plasmid for plasmid transfection. The mRNA is for mRNA transfection. And we also have a Viromer CRISPR for RNP delivery, which means you transfect the fully active Cas9 protein complex to a guide RNA into the cell, which means it's a protein transfection reagent. So, having a look at the possibilities on siRNA and microRNA transfection with the Viromer, I think the most important thing for you is maybe the table. So we have two reagents, like I said before, for siRNA and microRNA, the blue and the green, and they differ a little bit in their chemistry. So some cells prefer the green one, some cells prefer the blue one. In some cases, or in other cases, the cells like both reagents. And this is a list of different primary macrophages, also cell lines, monocytes, microglia cells. And with our reagents, we get more than, or in all these cells, we got more than 70% of knockdown efficiency. So for the plasmid transfection, we have the red and the plasmid reagent. And here are just some examples against some competitors for primary macrophages and also for the raw macrophage cell line. Sorry. Here I just show some examples for the plasmid transfection into different cells like the primary macrophages, cell lines, THP1 cells with different targets like CRISPR plasmids, luciferase reporter constructs, and so on. So if you are interested in this, take your time and have a look, deeper look into this publication. It's really, really interesting. So, and maybe some of you will say, okay, even with electroporation in my phagocytes, I got a pure expression rate. This could be due to the cytosolic defense mechanisms of these cells. So, think about it. So, the biology of these cells is actually to take care of us or take care of the host. They should not be manipulated so easy because they should take care of the immune system. And these cells have a lot of defense mechanisms against foreign DNA. And just as a short example, so there are different pattern recognition receptors, not just on the cell surface, always also inside of the cell, which is recognizing, as an example, double-stranded RNA, single-stranded DNA, double-stranded DNA, and also a lot of other things, just to be sure to recognize viruses and other things which should not be in the cytosol. So, and if you transfect these cells with DNA, and DNA is normally always in the nucleus and not in the cytosol, so they will try to fight against it. So if you transfect these cells with a plasmid, it could be possible that it's digested pretty fast. So the transfection is actually working. And we know so because we got a lot of feedback from our customers who compared the plasmid transfection against the mRNA transfection. And you see, for some cell lines, plasmid transfection is working really good, like for the raw macrophages, but for some primary cells like dendritic cells or the macrophages here, the monocytes here, mRNA transfection is leading to a much, much higher expression rate than plasmids, which is due to the defense mechanism. So mRNA transfection could be a real alternative for your overexpression studies. It's much, much faster because you totally skip the process of transcription. It is safer because there's no risk of genomic integration, and the production of the protein starts immediately. So here are some publications for the mRNA transfection into different phagocytes. Again, if you are interested, take a look. It's quite interesting what the people report here. And if you have the big question mark over your head and thinking about how to switch to mRNA transfection, so of course one possibility is to just buy the mRNA from different oligo suppliers. The other possibility is, so if you really see a pure expression rate using DNA, so the first possibility is to use plasmids with an SP6, T7 or T3 phage promoter. So it means you can easily use the plasmid that you bought from Origin before, take a little bit of it, linearize it, and use an in vitro transcription kit to do the mRNA production. You can also use PCR products and easily add the T7 promoter to the forward or the reverse primer. You can also use the cDNA using an oligo-DT T7 promoter. And the mRNA should be kept and should have a poly-A tail. But the procedure is actually pretty easy. It's like a PCR but faster. So you just put a buffer, the polymerase, and different nucleotides together and put it at 37 degrees for like an hour. Of course, you need to do a little bit of purification afterwards, but once you produce it, it will last for a really long time. You can store it at -80 and perform your highly efficient transient transfections. So as a conclusion for the cells of the immune system, Viromer can transfect successfully a lot of different phagocytes. So what we have reported back from our customers is certain kind of stem cells. We also have progenitor cells. We have like the HAP1 for the basophil cells. We have a lot of monocytes, primary ones, and also cell lines. We have data for primary dendritic cells. And we also have a lot of data for primary macrophages, primary microglias, and a lot of cell lines. And you can also go online and to our website and have a look at the immunology research page. There's an overview. There will be a table with recommendations, which reagent to use. You can see transfection or knockdown or overexpression efficiency. So you can see customer data and also the links to publications. So just to wrap it up a little bit, if you are working with lymphocytes, you definitely need to go for viral transduction or for electroporation. So Origin is offering a huge product portfolio for viral products, which you can use, and also expression plasmids to use in electroporation. But if you are working with phagocytes, and you can easily use polymer transfection reagents like our Viromer. They will efficiently transfect your phagocytes, and the biggest difference in using the Viromer reagents for the phagocytes is that you don't need to work in a security level 2 lab like you would need to do if you work with viral transduction, and they are definitely less toxic, more than less toxic than electroporation. So, in conclusion, this is our product portfolio, and Origin is offering a huge palette of targets, payloads that you can do, can bring inside of the cells. Different siRNAs, a huge bundle of different plasmids, also Cas9 mRNA if you want to go for CRISPR and use the mRNA reagent. And also a Cas9 endonuclease, so means the Cas9 protein and also synthetic sgRNA. And so Origin will be at the immunology meeting in 2 weeks in San Diego, the big meeting. So if you are there, stop by at booth 609. I will also be there, and we would be really happy to talk to you, to discuss maybe your problems, to talk about the needs that you have, and you can pick up your free Viromer sample. So thanks a lot for your attention. I'm happy to discuss all your questions.
Thank you, Bettina. Great, very nice and short this presentation to introduce us to a collections of reagents that you can use to deliver all kinds of payloads into your immune cells. So now we open up the floor for questions. So yeah, as we said in the beginning, the questions can be posted, if you have not yet tried, can be posted onto the console. Type that into the question box so that we can see that here and we can answer them in real time. So while we're waiting for other more questions coming in, Bettina, let me ask you a question. So those transfection reagents, does it work for the Jurkat cells?
No. No, because they are T lymphocytes and they don't do a lot of endocytosis, so no, not for the Jurkats.
Jurkat, we still need to go either virus
or virus, or electroporation.
Right. Yeah. Origin do offer virus solutions. So or plasmids or DNA, shRNA, we all have viral vehicles. So if you're truly dealing with something that no other solution, yeah, we have virus solutions as another resort. Oh, questions coming up. Okay. Sorry, I didn't see that. Okay, so now let's go over questions. Okay, here's a question, Bettina. What are the conditions for complex formation with Viromer and what is the success rate?
What is the what?
Okay, let me repeat. What are the conditions for complex formation? Basically, the whatever payload together with your Viromer chemical. What is the condition for the complex formation?
Oh, you mean the condition. So it's like with all the other transfection reagents. I just so you do it in an epi. So you dilute a little bit the Viromer and you dilute your plasmid, and then you put it together in an epi. You wait for the complexation like for 15 minutes, and then you put it on the cells. I just put this into the graph to make it a little bit more easy. And yeah, so it's done in an epi.
So basically, it's a single pipetting mixing.
Yes. Yes.
Okay. They also ask for success rate. Do you have numbers for that?
You mean for the transfection?
Mm-hmm.
So what we have here is more than 70% of a knockdown. We can also, let's say, go to the website. So this is what we have here is also, this is for the raw macrophage, just as an example. So you have 64% positive cells. Let me have a look. This is also the percent of transfected cells. This is the mRNA against the plasmid transfection. So we can also go to the website if we leave the presentation here, and I can show the cell database if you want to.
Okay. So yeah, maybe we after this we can add another material. You have your fact book, right?
Yeah.
Handouts that so many of the questions can be addressed in depth there. So right now we have this presentation and handouts that you can download. We can follow up with the additional material for those detailed questions. Next question, Bettina. Do you have any data regarding transfection efficiency of mRNA with Viromer in primary mouse bone marrow derived macrophage? Very specific. Because mRNA delivery in bone marrow derived macrophage.
So this one is definitely here, bone marrow derived macrophages, and the cell line, and also primary ones, mRNA transfection.
Perfect.
So your customer can have a look here, and This is also, this is not bone marrow-derived macrophages, but this is definitely. So, and you can always write me an email, and we can have another look at our publications and send you the several ones that we have.
Okay. All right. Another question: how long does it need? So, what your workflow, the transfection? How long does it need to do for the transfection? Your protocol?
I think about like 20 minutes. If you think it's a dilution of the Viromer, dilution of your pDNA. Normally you do it once. I mean the pDNA, and then you have a stock for this, and put it together. Wait for 15 minutes, and then you can put it on the cells. So let's say around 20 minutes, 15 to 20, something like this.
So it's just like
all them. Yeah, just like any other reagent.
Okay. Any toxicity issue?
So normally the Viromers are really, really gentle on the cells, so they are uncharged when they are complexed with DNA or whatever you can think of. So they are uncharged, so they will not stuck to antibiotics or the FCS in the medium, which means you can leave the antibiotics and also the FCS in the medium, which is not so stressful for the cells, so they don't need to starve or things like this. So normally the Viromers are really, really gentle on the cells, and of course, at a certain point, all the reagents will become toxic if you put too much on the cells, but normally not.
And so your protocol, does it recommend on the ratio between the payload and the
Yeah, so we have a really, really good optimization guide, which is, let's say, having a look at 4 different points where you can play a little bit on to get a better transfection efficiency, which is at first, of course, the amount on transfection complexes that you put on the cells. So you can, let's say for 96, well, you can put 5 microliters of the complex on it, or 10 or 15. So this is one point. Then, of course, there's playing a little bit with the Viromer to plasmid ratio. So if you have a bigger plasmid, of course, you would need more Viromer to get a better packing. And it's really simple. Like I always say, if you are 10 people who wants to go to the airport, we need more than 1 taxi. So it's the same here. If you have a really, really big plasmid, you need a little bit more Viromer to get a better packing. So this is a point where you can play on. Then, of course, you can play on the cell density. A lot of people don't do that and don't think about it, but if the density is too high and the cells are starting to get something like a contact inhibition, they will not divide anymore, and so the cell machinery is going down, so there will be no transcription anymore. So it's the same for primary cells. It's always hard. That's why it's a good idea to switch to mRNA. In that case, you see a pure expression rate with plasmids, but just in case.
All right. So move on. Let's get another question. How do Viromer compare with other transfection reagents? Do you have any comparative data?
Yeah, we have a lot of comparative data. So I will just show you something else. Should I go to our website to show?
You can go. Yeah, you can switch screen, and actually all audience can see. You're in control.
Okay. Just a second. Okay. So this is our website. If you come there, it's called the Viromer.transfection.com. So you have here all the reagents. Here you have the cell database, and here you have different research fields like immunology, metabolic research, and so on. So if you click here at Select by Cell Type, you will find the huge cell database. So in alphabetical order, you will find all the cells that people tested. You will find transfection efficiencies or knockdown efficiencies for siRNA, mRNA, and pDNA, and soon there will be also the CRISPR-RNP. And 4 dots always mean you have a knockdown more than 80%. 3 dots would mean 50 to 80%, 2 dots would mean 30 to 50, and 1 dot, just 1 dot, would mean under 30%. So if you're here, you can have a look at customer data. You can click here and publications. list of publications. So if we go to the immunology research field, here you can read a little bit, and then you will also find this cell database, but really just with immune cells. So you can go there, and just as an example for the macrophages, you see here great knockdown efficiencies, also great expression efficiencies with mRNA and also pDNA. And if you click on see data, then a PDF will open, and you will find a lot of data people reported back to us. So this is something you can take your time and have a look. And then also, if you click on publications for the macrophages, you will find all the publications that we have for siRNA and microRNA transfection into primary macrophages, raw macrophages, plasmid transfections, and so on. So take your time and have a look. There's a lot of competitive data because we always ask for that. It's also interesting for us how it is working against
right. So if any of scientists users, we also will be welcoming that you share the data with us, and we can post them on the website as well. So there's a lot of uncharted territory on cell lines. So we love to see your data with Viromers as well. Yeah. Okay, let's go on to the next question. So what MOI do you recommend for T cell using lentivirus? I suppose because there's still a need for T cell transfection, especially with the CAR T and the T cell therapy. So lentivirus right now probably is still the way to go for AAV. And right now for primary T cell, I don't really have a number to recommend. But for the T cell line like JERCAT, currently our website recommends 40, MOI number of 40, maybe even higher if you are using primary T cells. Okay, next question. We're interested in transfecting erythrocytes with RNA aptamer. Would you recommend Viromer?
Yeah. Which cell line was it?
The erythrocytes. Erythrocytes, I don't know whether they actually have phagocytosis. So the key is whether it has phagocytosis, right?
You said erythrocytes?
Yes, the red cells, red blood cells.
Yeah, red blood cells with aptamers.
Yeah.
Yeah, aptamers is it's a special thing. I would try it with the green reagent, definitely, because we got some really good feedback for aptamers with that.
Oh.
So I would try it with this. Yeah.
Okay, so the answer is the Viromer have a different color code. Viromer green is what King recommends. Okay, for aptamer. Okay, next one. Please explain the different transfection reagent for CRISPR methods. CRISPR methods. I think we talked about CRISPR RMP transfection in last webinar. They are posted on our website. But I think, Bettina, why don't you give a quick answer for this one? What are the transfection reagents for CRISPR methods? Okay, a really quick answer. Otherwise, the person can have a look at the webinar. So the biggest difference, there are a lot of possibilities to go for CRISPR. You can do it with a plasmid, Cas9, guide RNA, everything on one plasmid, or it's on two different plasmids. Then you can also do it with a Cas9 mRNA and a guide RNA, and you can do it with a RNP complex, which means it's a ribonucleoprotein complex. And as you can see here, the biggest difference is when you have a plasmid, it needs to be brought inside of the cell, then it needs to be brought into the nucleus for transcription, it needs to be brought outside again. So then you have the Cas9 mRNA and the Cas9 guide RNA. The guide RNA is ready, the Cas9 needs to be translated into the protein, and then the protein needs to find the guide RNA and assembly, and then it needs to be brought again to the nucleus for genome editing. So if you use a preformed ribonucleoprotein, it's ready from the first second to work. It will be brought inside of the cell and then it goes directly to the nucleus to do genome editing. And also for let's say if you do it with a Cas9 mRNA, I don't have a picture at the moment here, but you skip the process of transcription. So depending on the cells you're working with,
you have a lot of different possibilities to go inside. Some cells are really happy with the plasmids, or a lot of cells are really happy with the plasmids. Some other cells are more happy with the preformed RNP complex. It has less off-target effects. And so Origins offer all the different possibilities that you can think of. So they offer guide RNA, they offer Cas9 mRNA, also the Cas9 protein, and also a lot of different CRISPR plasmids. So yeah.
So I'm trying to, because there's still questions, I'm trying to be able to finish more. So basically the short answer to that question is there's multiple ways of deliver CRISPR machinery into the cell. And everyone, we have a solution to that. And please come to visit either Lipocalyx website or Origin website. And we also have a recorded webinar for that question, the whole thing in detail addressed. Also, you can send an email to us. Also, we can send you the link. Okay, sorry to interrupt you because I do have additional questions I have to address. Okay, next one. Will virus transfection or Viromer transfection activate immune cells? So that's a big question, huh?
So it's a question about the immunogenicity. So to our knowledge, I would clearly say no. So we had some recent publications lately where people made their own mRNA. So it was not a clear test for the Viromer, but the intention was to test their own mRNA they made. So the cells made mRNA for testing them for an immune response, and together with that experiment, they of course also tested the Viromer for an immune response in THP1 cells or primary macrophages, and the answer is clearly no. So if you're interested in that, you can write me an email and I will forward you the publications to that.
Okay. That's a great. Yeah, the publication probably have a lot more detail. Okay, then the next question. So compared to electroporation, what are the problems? Basically, what's the advantage of Viromer compared to electroporation?
Of course, electroporation is there's a huge toxicity because a lot of cells die when you have these electrical or when you put the electricity on the cells. So, and the electroporation is just disrupting the cell membrane for a really short time, so the plasmid can go inside of the cell. And the Viromer is gentle on the cells, and the Viromer is just packing the plasmid you want to bring inside of the cell, and the cell is taking up the transfection complex on its own. So this is the biggest difference between electroporation and normal transfection.
So basically, you don't bring trauma to the cells by unlike electroporation, you bring big trauma.
Yeah, that's true.
Okay. So next question. How long do the cell need to incubate before it starts to expressing the desired protein with mRNA transfection?
You mean the question is how long does the cell need for expression when you use mRNA?
Right. After mRNA transfection, how
long? It can start as early as 2 hours after transfection. It's pretty fast. 4 hours, 6 hours, you can see already the whole cells lightening up. So it's pretty fast.
So that's the advantage of using mRNA. One of the advantages, the really expression results you can see. Okay, next question. How is your formulation compared to lipofectamine? I think this is a chemist question. You know
the chemistry question, So the Viromers are polymer based, like I said before. Lipofectamine is lipid based, so it will always influence with the cell physiology. Because if you bring a lipid to a lipid double layer, like the membrane is and all the other membranes inside of the cell, it will always interact with the cell physiology. And the Viromer, since they are polymer based, will not do that. So it's a definitely big plus point for the Viromer.
Okay, another question, very relevant. Is this transfection efficiency similar between adherent cells and suspension cells. Can you work this one with suspension cells?
Yeah, definitely. Let me have a look. You can also sort the cell database for suspension cells. So these are all the suspension cells. For siRNA, it's working perfectly. For pDNA, like when it's immune cells, like I said before, and other ones, it's a little bit harder. So mRNA is working always better. And for suspension cells, it's let's say it requires a more optimized protocol. And since these cells grow in a higher density inside of the medium, or let's say of the bell, it's always a little bit harder to transfect them.
Okay. Next question. Does the use of Viromer require media change? Is this
no?
toxic?
Like I said, normally no. If it's not binding to the antibiotics and it's not binding to the FCS, so normally no. But we always say if there is a possibility you see toxicity, we always let's say suggest to change the media for hours after transfection, but just if you see toxicity, because normally you say what the cells didn't take up after 4 hours, they will never ever take up. So the whole process is done after 4 hours. And media change is just suggested when you see toxicity.
Okay. Okay, next question. Can it be used for in vivo?
I knew that this question will come. Yeah, you can, well, there are Viromers for the use of in vivo. I would not recommend to use these in vitro reagents, which I showed you, for in vivo, because they don't have the, let's say, the purity and the safety to use in animal experiments, like mice or rats or so. But there's another part of the company, of the Lipocalyx company, which is really dealing with this, providing a lot of different Viromers to go in vivo. So if you're interested in this, yeah, just contact us and we can talk about it. But I would not recommend to use these in vitro reagents to go in vivo, just for the safety of the animals.
Okay, so there's another question that beyond the 293 T cells, do you recommend the different cell lines for efficient lentivirus packaging using Viromers? For this question, normally because Hex 293T is easy to transfect, so the lentivirus production is usually high titer. So normally people just use Hex 293T cells. So with a transfection reagent, Hex 293, you can use Viromer or any basically any other transfection reagent. But Origin do have a high efficient lenti packaging kit with the transfection reagent already included.
Okay, good. I think we are almost at the end of the question. I see one question regarding a particular cell line called U293. For that question, the data from just Bettina showed on the website actually says using that one works in Viromer blue. So that you can check out that PDF, that web page on the Lipocalyx shows this particular cell line works well with Viromer blue.
But the blue is siRNA. It depends if the customer is transfecting plasmid or siRNA.
That one has been tested in Viromer blue.
The blue is only for siRNA.
Okay, so the other one is not tested. So you need to test currently the data has been tested for Viromer blue works beautiful. Okay, I think that we are at the end of today's webinar, and thank you so very much. Again, you're always welcome to reach to us through email. And we will follow up with another email with a survey.
Oh, yes. There is a survey after the webinar. You can request the Viromer samples. So we will know that how many samples we need to bring to the meeting. and you can pick the sample up.
Yeah, on your exit, we also want to do a survey and see what's your response to this, and we love to hear from you. And if you have additional, or your colleagues have additional requests, feel free to send to us as well. We love to address your questions. Okay, thank you so much. Thank you, Bettina.
Yeah, you're welcome.
And thank you everyone for being with us in the past hour. Have a good day, good afternoon, and good night.